everyone, I am Dr. Sonal Jain. In this series of forensic science lecture, today we will be covering a topic related to wildlife forensics. We will be learning about techniques which are used in the identification of different wildlife samples. We will be studying about the various wildlife samples, how to handle those samples, the preliminary techniques to analyze them, the confirmation techniques which can be used to analyze the wildlife forensic samples, and the stable isotope studies which can be done on those samples. To start with, we might find the various variety of wildlife samples on the scene. For example, tissue, bone, eggs, blood, feces, hair, tooth, etc. So these variety of samples, they can be taken or they can be found at various scenes. For example, let's imagine a case of poaching where any species which is going to be extinct or on the verge of extinct or already extinct, if there are certain species, such can be used or they can be transferred for various selling purposes. For example, there are certain species of snakes which are being transferred or smuggled for their skin. So based on that, or to identify other wildlife species, we can use this sample for their analysis. To handle the samples, first of all, we have to avoid the contamination of the sample by the collecting officer or by the officer, other person, where which the animal sample is associated with an investigation. The sample collector should always wear clean gloves to handle and prepare each sample. Disposable sampling equipment should be used. So one time use, and after that you have to dispose of it carefully. So we have certain wildlife samples, for example meat, organ and hide samples. Depending upon what kind of use these animals or these wildlife sample can be used to, they can be processed and shipped using preservative buffers. We have to label the sample tube marker using a permanent marker. We use clean latex gloves to handle the sample. With a clean scalpel, shave off a small portion from the outside of the tissue and discard. Then we cut a piece of tissue approximately the size of 1-inch spaghetti noodle, for example, which could be around 7.5 mg from the inside of the tissue. So that is how we do the collection of the tissue samples in the wildlife cases. Then we add the sample to the buffer. Then tubes can be shipped at room temperature. Excess tissue samples should be kept frozen at the respective facility. The reason being that whenever we need the sample again, we can take from the respective facility. Then we come to the preparation of wet blood stains. So they can be found at the killing site. There are certain animals, their poaching is banned, but the people, they kill those animals for their own benefits. So at such sites, we can find certain samples which are commonly found on leaves and rocks. The blood is commonly found there. If the stain is large enough, we sample the blood using a dry swab without contaminating it with leaf material or dirt. We air dry the swab for few minutes and then place into the sealed tube provided and ship at room temperature. But if the quantity of blood is less, then we save the leaves and place the blood stain attached to the organic material into the desiccant tubes. The sample will be stored and shipped dry. So in the cases if the blood sample is literally less, then we send it directly. But in the cases, if the case is old, there could be dried blood stains which can be found at the scene. So samples located on materials such as clothing, knives, gun, etc., they should be shipped at room temperature in a well sealed package. Blood stains may have to be cut or isolated from the material. If blood stains are located on an object that cannot be shipped, we collect the stain using a clean swab moistened with phosphate buffer saline. We dry the swab and we always air dry it and ship it to the laboratory in a well sealed tube or container. So that was about the blood samples. But the major kind of samples which we come across in the wildlife species are the hair and feather samples. Samples should be kept air dry and shipped at room temperature in envelopes or a sealed plastic bag, depending upon which species hair or feather it is. DNA is usually isolated from such samples from the root of the hair shaft, and therefore it is extremely important to have hair samples with root, because then we can identify the species from the DNA extracted from the root of the hair. If the meat samples are present at the crime scene, these samples for quantification such as sausages or cooked meat should be shipped in a cooler with ice. There are certain places where a particular species or animal eating is banned, and if the forensic expert come across such a case, then the cooked meat should be directly shipped to the forensic science laboratory. The entire meat sample should be submitted to allow an accurate assessment of the species of origin. We can freeze the sample and ship in an ice-packed sealed cooler. So, in the cases of the whole blood samples, blood from live trapped animals can be collected on blotting paper using the needles provided. We can draw the blood from the specimen, blot blood from the animal onto leaflet inside the insert. We air dry it for the 5 minutes and then replace, insert into the envelope and then we transfer it. The major thing here is we do not have to seal the envelope. Then place the envelope into a sealed plastic bag with a desiccant pouch and the blot should be shipped at room temperature. The blot which is being shipped at room temperature, if we are using a vacutainer, the remaining blood samples should be frozen and kept at the respective facility. One other major case where the samples are being found is in the form of bones. Bone samples could be from teeth, antlers and carcasses that can be used for DNA analysis. Most often bone samples are provided from historic samples. The samples should be collected in a sealed evidence bag and transported at room temperature. Whenever we are collecting the fecal samples are majorly being collected, we wear the gloves, we place the feces into a collection tube, do not fill more than halfway. The surface layer containing the target DNA therefore ensure that the outside of the feces is sampled. We close the lid, place the tube in the evidence bag, seal, label and send it to the laboratory. Generally in the cases wherever we do not come across a very wild animal, in such cases the various forensic wildlife experts, they identify through the feces or the amount of feces how many animals of that particular breed they live in that particular area. So, fecal samples are very important to collect and in major cases the DNA extraction can be done through those fecal samples. So, whenever we have collected the samples, there are certain preliminary techniques which can be used to know to whom those samples belong to. These preliminary techniques, if we talk about, they are a non-definitive screening test to give an indication that something might be present of our interest. The positive results indicate the need for the further test. So, supposedly if you are able to identify a species based on the preliminary techniques, then we do not have to go to the confirmatory techniques. The reason behind that is the confirmatory techniques are quite expensive. But if you are able to make sure about the species to the preliminary level, then it is better to use the non-definitive screening test, which are the preliminary techniques. So, the first technique which we use is the microscopy. The microscopy, the use of the microscope can be dependent upon what type of sample it is. For example, generally for the hair, the species can be identified using the optical microscopy, or the feather can be read, or it can be studied under the optical microscopy. But there are certain samples, for example like the bone samples or the eggshell samples, they have to be studied under the electron microscope. The reason behind that, through the electron microscope we can see the various layers which are present in the bone or the eggshell samples. So, we have to slice the sample and we have to place the thin slice if we are using the TEM, that is the transmission electron microscope. But in cases where we are using the SEM, that is the scanning electron microscope, we can directly keep the samples in the sample holder of the microscope. Now, when we talk about the microscopes, the optical microscope often referred to as the light microscope. This is a type of microscope which uses visible light and a system of lenses to magnify image of the small samples. So, the sample is kept over there and the natural light or the artificial light can be used, and we can, depending upon what kind of magnification we can put, for example, it could be 4x, 10x, 40x, 100x. So, we can magnify the sample up to that level. In fact, the microscopy can help us to identify different various species of hairs. A dog hair inside a microscope and a cat hair inside a microscope will appear different, and you can identify the species with that. Now, coming to the optical microscope, they are the oldest design of microscope. Basic optical microscopes can be very simple, although there are many complex designs which aim to improve resolution and the sample contrast. So, depending upon, we can use different filters and we can identify which filter to be used for which particular sample which we have to analyze under the microscope. As I have given the example that even the species identification of human hair or the animal hair or the differentiation between different animals can be done using an optical microscope. In fact, if we are taking the feather, which is just the animal or the bird hair only, we can easily identify the species using the literature which we are having. So, in the microscope, a clear distinction can be made between the animal hair and the human hair. Scanning electron microscopy and the transmission electron microscopy can be used in such cases where we have very thick samples. For example, let us talk about the cases of eggshells, where the samples are quite thick and the light cannot pass through them. So, in such cases, the scanning electron microscope is being used. The scanning electron microscopy is not similar to the optical microscopy. In optical microscopy, we are using light for the analysis of the samples, but in the scanning electron microscopy, we are using a beam of electrons to identify or study our sample. A SEM or scanning electron microscope is a powerful magnification tool which utilizes focused beams of electrons to obtain the desired information which we have to get from the samples. So, these are the very high resolution 3-dimensional images produced by SEMs. They provide the topographical, morphological and compositional information which makes them invaluable in a variety of science and industry applications. So, the avian eggshell, if we see under the SEM, different layers can be observed, which can help us in telling that this eggshell belonged to some avian. So, we can see different layers under the magnification and we can identify the species using that. So, what is the positive point of using these preliminary techniques? The best positive point about these preliminary techniques is that we do not have to destroy our samples. In microscopy, the samples can be directly put and they can be directly visualized. When we are going for the confirmatory techniques, those confirmatory techniques require the destruction of the samples. Now, X-ray diffraction is one of the other technique where we can use and directly know the composition of the particular sample we are handling with. For example, let us talk about any bone or let us talk about any eggshell through which we can know what is the basic most widely used technique for the identification of unknown crystalline material. We can get to know which minerals are present, which inorganic compounds are present, and we can also determine unknown solids, which is critical to study the composition of samples and measurement of sample purity, as well as we can get to know how much degradation in the sample is present over there. For example, if we are putting a sample of different bones of fish bone, frog bone, pigeon bone, we can see that the peaks in the XRD, they are different and they are varying differently. So, we can identify any sample while we are using the X-ray diffraction and we can match the graph with the literature available outside and we can see that this particular species shows what kind of signals. Then we have electron backscatter diffraction, which is the EBSD, where we use the technique of backscatter, and when our sample is put in the EBSD machine, then we can see the original composition of our samples. So, this is a microstructural crystallographic characterization technique to study any crystalline or polycrystalline material. The negative of this technique is that only crystalline materials can be studied through EBSD. This technique involves understanding the structure, crystal orientation and phase of materials. Like we can see the EBSD showing difference in crystal structure of chicken and ostrich cell. For example, if we say that through scanning electron microscope we have identified that this particular eggshell belongs to avian species, but the EBSD can be help us in knowing that this particular eggshell belongs to which particular species. We can say the left hand one belongs to the chicken and the right hand one belongs to the ostrich species. While we are conducting these, there is one more technique which is confocal laser scanning microscope. This microscope is being used to see inside the DNA hotspots present in the samples of eggshells or bone. For using this technique, we use a variety of dyes and the sample is dyed using those dyes, and if the DNA is present inside the samples, then we can see the hotspots of that. So, this confocal laser scanning microscopy is also known as LSCM, which is a technique for obtaining high resolution optical images with depth selectivity. The key feature of confocal microscopy is its ability to acquire in focus images from selected depths and a process which is known as optical sectioning. So, when we are doing the CLSM, the images are acquired point by point and reconstructed with the computer, allowing three-dimensional reconstruction of topological complex object. So, when we have a three-dimensional reconstruction, then one can see the inside. For example, while we have we use the dye hex 33342 to dye a sample of eggshell, we can see the different hotspots which are present there because that dye dyes the biological specimen which is present in the eggshells. So, if we can clearly mark the area where these hotspots are present, and on that basis we can identify through which that particular area can be used to extract the DNA. So, after we get to know from the CLSM analysis that where the DNA is present, we can further go for the DNA extraction. So, here I want to point out that depending upon what kind of sample you have encountered in forensic examination from the wildlife, your DNA analysis will be different. The DNA analysis will be dependent upon various things: what kind of sample is it, through which species it belongs to, and how to go about it. The procedure involves the sample preparation for DNA extraction, DNA isolation and quantization, PCR amplification with species specific primers, and DNA sequencing through which we can go for the phylogenetic studies. Now, here when we are talking about the sample preparation, depending upon what kind of sample you are having, whether you are having a hair sample, whether you are having a feather, whether you have DNA of extraction you want to do from the bone or the eggshells or the feces. So, the DNA extraction protocol will vary according to the sample which is present over there. After you have used a specific protocol for a particular sample, we move to the DNA quantization. So, quantity of DNA can be estimated by calculating the optical density using spectrophotometer. We can use a UV visible spectrophotometer as well as we can use the nanodrop machine. So, the DNA quantization is done after the DNA extraction to know how much quantity of DNA has been extracted from the given sample. DNA quantization will also vary from sample to sample. For example, if a bone sample is there, the quantity of DNA would be high, but if the hair is there with the root, the quantity of DNA would be very less. So, we need a lot of strand along with the root to extract the DNA from the hair samples. But if the root is not present with the hair, then we cannot extract DNA from such hairs. So, depending upon the sample which is being provided to you, the quantity of DNA will also vary through that. After the step of DNA quantization, we move to the PCR amplification and DNA sequencing. Here in this part, we have to use the PCR, which is polymerase chain reaction. For the specific reaction, we amplify specific gene and we sequence the PCR products. Then in the next step, we align those sequences with the published sequences which are available on various literature sites, for example, NCBI, PubMed, etc. And then we analyze them with different phylogenetic softwares. So, for the first step, when we are amplifying any specific genes, what we need? We need species specific primers. For example, the primers which will work for a particular species of pigeon, they will not work any species of other avian or birds. So, depending upon from the previous data which you are having, what kind of species could be that, we have to make species specific primers for conducting PCR. PCR, we have different stages where we basically the DNA which we have extracted, we make multiple copies of it. The multiple copies can be made using various steps of denaturation, annealing and extension. In these steps, whatever DNA is there we can make the million copies of them by multiplying it exponentially. So, this is the step, or this was the trademark, or this was the invention which has helped us for the DNA extraction and which has helped us further for the quantization and making copies of DNA. So, PCR basically make the multiple copies of the gene in order to sequence it. We use the primer extension reaction for amplification of specific nucleosides in vitro. So, everything what we have done is being done in vitro. After PCR, we have to go for the gel electrophoresis. Why it is required? We need to know whether the samples which we are making copies of are the right copies are being made or not, because they will show their band at a specific location depending upon the size of the band. Now, how this band size comes? These band size comes by the way we are using the PCR specific primers, and these specific primers will amplify that particular region, and we know that how much particular a specific band size will come in gel electrophoresis. So, after the gel electrophoresis, we have to go for the sequencing of the DNA. There could be automated sequencing where we use the capillary array contains polyacrylamide gel. We use capillary passes through a laser. Each dye fluoresces a different wavelength and when excited by the laser, and the fluorescence is detected by the CCD. This way the capillary gel electrophoresis occur. So, the bases which are present there, they are being given different dyes, and depending upon what kind of fluorescence occurs, we can see how the DNA is being sequenced. Fluorescence are processed into an electrophotogram, and then we make the base calls. The base calls are made by sequencing software which can be analyzed manually further. So, we can see in capillary electrophoresis, the base calls is being given on different colors of the dye. For example, we have used yellow, blue, green, red over here, and then we can see upon depending upon the base calls what is the sequence of DNA we are analyzing. Now, the sequence which we have produced from the various sequencing platforms, we have to align them using various published sequences. The published sequences are present on various websites, for example, NCBI, PubMed, Protein Sequencing Database, Nucleotide Sequence Database and Genome Database. So, whatever sequence you have procured, that sequence, let us take an example of NCBI, you will put that sequence in the NCBI website and then you will blast it. Blast is an option over there. So, after you will blast that sequence, your sequence and the sequence which are already present on the NCBI, they will show a match. Whether it is a 100% match, 70% match, 50% match, 20% match, depending upon what kind of sequence do you have you got from your extracted DNA and further your sequenced DNA. Those can be matched depending upon the sequences which are already published on the NCBI. And if you have a new research, then you can put your new sequences into the NCBI and they will give you an GenBack accession number. So, after they are aligned with various species and phylogenetic trees are constructed. This type of trees will tell you how close these species are up to each other. So, the phylogenetic analysis will help us in knowing that particular species, the closeness with different species, and will help us in directly knowing whether our species is being closer. For example, the example is being given of the ostrich, which is more closer to the kiwi, emu, and which is less closer to the other birds. So, this phylogenetic analysis help us in knowing about your species and closeness with the other species. That is how we do the phylogenetic analysis. So, to conclude, I would say that right from the correct evidence collection to the DNA extraction and moving towards the phylogenetic analysis, we can get to know what species are present at the wildlife crime scene samples. Thank you.